Oh, well, to begin, thank you for staying to the end of this session. I'm Carlos Angel, as it has been said, and I will be presenting the first part of my work on my PhD. So we all know that two-dimensional structure of the genome is studied with high C data. And high C data is represented with these matrices where the genome is fragmented in equally sized beans and the intensity on the matrix is represented by how many times these fragments are contacting, and the coverage of these matrices is really important because it allows us to identify these um, these regions, such as enhancers, that are gonna regulate a given promoter in a given time. However, it is really difficult to identify these interactions in high C data because they require like a big coverage, and even like up to 10 kilobases high C resolution, they are like pretty difficult to identify. And therefore, also it's super expensive to achieve high resolution. Some of the power laws and some previous studies have reported that to identify most of the loops in a genome, we will need 25 billion contacts in a high C matrix. However, this is prohibitive, and also, although some of the consortia out there, such as the ENCODE project, is recommending 3 billion contacts, only 35% of the loops can be identified with statistical significance at this with this amount of contacts. Therefore, most of the data sets in the literature are of low resolution, and therefore there is a pressing need to develop algorithms that allow us to go from low resolution to high resolution high C matrices. So we are motivated by the use of a physics-inspired folding approach that models chromatin as a polymer to infer missing interactions. And this is similar, and I want you to think on how a pretty simple method to fold a shirt can end up in a like perfectly folded shirt. So that led us to develop what we call pC-SAC, that stands for probabilistically constrained self-avoiding chromatin. And this is an adaptive importance sampling technique with sequential Monte Carlo that is designed to produce high resolution, high C matrices by constructing chromatin chains based on the low resolution frequencies. So I'm going to give an overview of what pC-SAC does, and it begins by taking low resolution high C interactions and derives a set of constraints of these interactions. Then a subset of these constraints are chosen, and we start constructing these chromatin chains one monomer at a time by following two important constraints that are the collision and the distance constraints. I will explain more on this later, but we will construct these chromatin chains, and then this process will iterate over and over again by selecting different constraints for each chain being generated. And at the end, we will possess complete set of chromatin chains, generating an ensemble of chromatin chains that will be translated into a high resolution high C matrix. The way that pC-SAC achieves this is that the low resolution interactions or the input can be projected into high resolution beams by these ranges J and K that actually just maps the low resolution to the high resolution interactions. So in each of the iterations that we are generating a chain, each of these interactions in high resolution beam is going to be chosen, and at the end we're going to have ensemble of chromatin chains with perfect frequencies on a high C resolution matrix. So the main purpose of pC-SAC is that we want to generate an ensemble of chromatin chains, and this ensemble can be defined as the um, just the collection of n chromatin chains, and a chromatin chain is just the collection of n nodes, and the position of each of our nodes is just a set of 3D coordinates. Therefore, the configuration of any given chain can be defined as the collection of 3D coordinates of end nodes. So the main goal, as I previously mentioned, is that we want to generate an ensemble of chromatin chains, but this ensemble has to maximize a target distribution, which is a distribution where all of the created chromatin chains will satisfy all of the imposed interactions. However, this is visually impossible. Therefore, pC-SAC relays in sequential Monte Carlo, and this is that in each iteration for a given chain, we take a subsample of these interactions and we generate what we call a trial distribution. Then, by following this trial distribution, we try to construct these chromatin chains, one monomer at a time, by following these two simple constraints, the distance and the collision constraint. The collision constraint just takes into consideration that any of the monomers added into the partially generated chain cannot overlap with any of the previous nodes, and the distance constraint just takes into consideration if a node has to be interacting with any of the previously positioned nodes. So going more into details of these two properties, the self-avoiding property can just be defined as an indicator function that will evaluate if two monomers should be if two monomers are colliding or not. And in this case, if they are not colliding, then we'll attribute a 0. And if they are colliding, we attribute a 1 because we don't accept any collisions. And the distance property is something similar, and it's an indicator function that evaluates if two monomers should be interacting in the chromatin chain. So we follow this iterative process where in each of the iterations we generate a chromatin chain, and then all of the interactions that are satisfied in a given chromatin already generated are subtracted from the initial target distribution, and then we update the target distribution to generate a new trial distribution. Then we generate more chromatin chains, and this process goes on until all of the initial interactions are completely satisfied. So moving on, how we validated pC-SAC, the first thing that we did is that we tried it in synthetic data, and we generate a synthetic chromatin Hi-C matrix with a defined pattern, and we downsample in different rates using that as an input for pC-SAC, and then we just reconstructed the Hi-C matrix. So we evaluated the performance of our algorithm by using the precision and recall. As you can observe in here, in all of the downsample um, percentages that we use, pC-SAC achieves pretty good results from up to 0.96 precision and recall. Moving on, we also evaluated our algorithm in experimental data. And for this, we randomly selected genomic regions that go from 2 megabases to 3 megabases. And we applied a similar strategy. We downsample in different rates, and we reconstructed the high resolution matrices using pC-SAC. To evaluate the performance, we use different types of metrics, such as correlation ones, image quality, and error matrix. As you can observe in here, pC-SAC in all of the metrics, by taking the average of all of the tested regions, achieve pretty good results, indicating that actually the reconstructions that we generate are pretty are not identical, but pretty close to the original experimental Hi-C matrix. Also, because we wanted to provide a pretty robust benchmarking, we compared pC-SAC performance to all of those deep learning algorithms out there that serve for the same purposes. And we observed that in all of the previous regions and previous metrics that I mentioned, pC-SAC either outperform all of the methods or has pretty comparable performance. However, here is important that we make this disclaimer that pC-SAC is the performance of pC-SAC depends a lot on the amount of input constraints that we are giving it. So it will excel at a specific loci reconstructions, but deep learning methods are a pretty good alternative if you want to make studies at the genome-wide scale. Also, we show that our method excels not only in reconstructing short-range interactions, but also in long-range interactions. something that deep learning methods actually fall behind. And most importantly, pC-SAC not only can reconstruct these high-C maps, but can identify new biological interactions that are actually not present in the experimental high-C matrix. To understand if these interactions were actually meaningful, we perform a cis regulatory element analysis, and we define the interaction as a true positive if there was an overlap in both of the anchors involved in an interaction, and a false negative if there was non-overlap. When we analyze all of the previous mentioned regions, we observe that the mean pre-enrichment on all of those novel pC-SAC interactions is almost 88%, which is comparable to what the best deep learning model out there achieves and even surpasses it. When we tested for statistical significance, we identified that almost all of our regions possess significant interactions, significant novel interactions. Uh, Lastly, we also tested the power of pC-SAC, and we wanted to test if pC-SAC could breach different types of methodologies. And for these, we use micro-C data that as a low resolution inputs. And when applying pC-SAC, we were able to reconstruct what is called RCMC. And when we compare the RCMC matrix to our pC-SAC reconstruction, we observed that we almost achieve 0.95 of Pearson correlation. Therefore, pC-SAC successfully breaches these two methodologies that completely different from each other and have completely different methodological steps. So just as a concluding remark, I have presented pC-SAC that reconstruct 3D chromatin ensembles from low resolution high-C data using sequential Monte Carlo and achieves up to 95% of accuracy. We discovered novel high-C interactions that are actually biologically relevant, and we can breach experimental techniques such as micro-C and RCMC. And the important thing is that we have to think as pC-SAC as a good alternative to study low size of interest. With nothing else to add, I would like to thank my amazing advisor, Dr. Gamse Gursoy and Nargis Elamori, to help us with the synthetic validation of the algorithm, and all of my lab mates, the G2 lab, which have been like a super great team to work around. Um, so yeah, if you're interested in using pC-SAC or learning more about the method, it is available in Nucleic Research. So thank you.
Great. Thank you for excellent talk and pSAC. And actually, I think it's the first one that doesn't use deep learning approach for enhancement. We're just curious. In the comparative table you showed earlier, I was wondering the downsampling ratio. I know pSAC could do across different downsampling, but the result, if the other methods were compared at a particular downsampling ratio, and also
Yeah.
wanted to know, given that pSAC does point of interest as well, um, how you had to create the data, the training data sets, all those other methods were trained on.
Okay, for the first question, can you repeat it?
Yeah, for the first question.
Oh, the downsampling ratio. So the downsampling ratio, so we perform the benchmarking using all of different downsampling ratios. The ones that I presented, it was 1/16 of the original coverage of a high-C matrix, but we presented the supplementary data, all of the results compared with the other ones. So the results are comparable in all of the downsampling ratios, except when you use just 1% of the initial data.
Okay. So then for the training, then in the other for the other methods, because how did you have to do that? Especially, was it cell to cell or same cell training?
So, oh yeah, we use exactly the same cell line with all of the deep learning models and with our method.
Or training and testing, right?
Yes.
So the other ones were the other training data sets you use for that ones, were they like specific chromosomes you use? Because pSAC doesn't, it does.
Oh yeah, we don't do training. Like we use the same chromosomes for all of the training from the other methods.
Other questions? I had one question, which is when you're sampling from the distribution, you propagate your constraints iteratively in one direction. I wonder if you tried actually doing it in the opposite direction and if you get different results.
Oh, actually, yes, we generate chains in the other direction, and we get exactly the same results. So,
Interesting. So then the is it guaranteed that would happen because is the folding energy, the energy function that you're using, guaranteed to be symmetric?
oh, well, I actually don't have a question for that, but the results seem to imply that it's guaranteed that it will happen. But
Okay, good. Anything else? Maybe folks are hungry for lunch, which is what's
are.
next. So thank you again.
Thank you.